that I'm going to talk about marine, and I'm the most inappropriate person to talk about it for two very simple reasons. First, I don't know how to swim, and I will be talking about swimming. I don't know really about swimming at all. Like, I can't swim. Second, I'm a vegetarian, and I'm going to talk about planktons, zooplanktons, fishes. Okay, I'm going to show you as an example. So I assume your background is physics and all physics. Okay, or engineering as well. Okay, very good. So you know everything. So what I'm going to talk about is collective dynamics of active agents, and you will see what I mean by active or activity. And since this session, I guess, is about marine examples, I mean, one has to take examples from the marine world. So I will take two examples from the marine world, but then I will convert it into a typical physics problem. Okay, simplify to the extent that the oceanographers, marine biologists will start hating me, but that's what we do, and we will try to get some sense out of it. Okay. So whatever I'm going to present is in collaboration with Anupam and Akshay, who will be here, I think, this evening, and two of my students have worked on it. So Deepto is a joint student with Anupam. So what are these two examples? These two are collective nutrient search by marine bacteria. That's the first example I'm going to consider. Second example is the orientation dynamics of phytoplanktons in oceans. And these two are linked to some extent, and you will see. Okay, so how many of you recognize anything from this? This is a simple or a complex food web. Okay, so you have in the ocean phytoplankton, zooplankton, fish. Okay, then you have microzooplankton, bacteria. Okay, so this is one classic food web, and this is another, which is also called microbial food loop. So where what we would be doing today, so looking at this, okay, and something about this, phytoplanktons and bacteria. So for us, these two will be the active particles, and these thrive in the marine environment. So that's what marine would be. These two problems for these two objects. Okay.
So, can I ask, because I'm not sure, can you tell me how to read this food web? Is it what? So the phytoplankton eats the plankton?
Basically, you know, I'm not a geologist. I would just say who eats what.
Yeah, exactly.
So in that sense, that is one chain, and then you can have this chain where this is extremely important. This is all organic carbon, and these guys play a very important role.
So who eats what?
Let's say this way.
Oh, this way it's eating.
This is one.
Okay.
Then you have this one based on size. I mean.
Oh, I see.
Okay. So higher level of trophics as you go, and people might hate bacteria, but bacteria are extremely important. You might hate algae, but algae are extremely important. 70% of the world's oxygen is there because of this. So we survive because of these phytoplanktons. Okay, so now let's have a closer look at these microbes. So I mean, there are a variety of them, but I will focus only on this guy, and again I will just reduce these to an active particle just for sake of example. You have Escherichia coli, okay, and you have Pseudo-Altermonas haloplanctis, okay. So these are heterotrophic bacteria. So does anyone know what do I mean by heterotrophic?
No.
How many of you cook for yourself?
Have you cooked for yourself?
You cook for you? I also. But do you produce, are you like plants, water plants? They can cook for themselves. So these bacteria, they depend on outside food source, okay? So that you can see from here. So you have phytoplankton. When it dies, decays, undergoes lysis, these bacteria feed on it. So that's heterotrophic bacteria for you. And whatever
This
is
means those who depend on the
others. So plants are autotrophic for that matter. So you're eating something, right? Nutrients. And what are nutrients? Chemicals, right? So now if this bacteria has to eat, first it has to find where those nutrients are, right? So it should have some mechanism to find out that food, right? So how do you find out food? Let's say something, he's cooking something very well. How do you find that? Smell, right? And what is smell? What is smell? Chemotaxis. Right? And if he's cooking something horrible, you would also like to run away, right? So those two, I mean, so there is some chemical. Okay, chemical and these bacteria, mess food. These bacteria are trying to locate. So there is a chemical substance, which could be nutrients, which can be poison as well. So these bacteria have the ability to sense sometimes the chemicals or sometimes the gradients of those chemicals. Okay. Now, the nutrient is in the marine world. That is it. It's in water, and water's ocean, right, or lake, right? So, and there can be a flow there, right? So the flow may frustrate the chemotaxis. Let's say you're trying to, find the source where the smell is coming from, and there's a strong wind in a different direction. So you might get confused. So flow may frustrate chemotaxis, or there can be a shear trapping, if you know shear flows, trapping, which can prevent from turning these bacteria in some gradients. So in the past, many experimental works have focused on the motility of these works. You can look at some of these examples. Okay. Then, what is chemotaxy? Ability of an organism of microscopic dimensions to sense and respond to macroscopic chemical gradients. Okay. So I was talking about a lysis event. So here is a single, let's say, phytoplankton, and the phytoplankton is like, let's say, dying. Lysis is happening. Things are coming out. So I will just play this video for you. See, these smaller ones are the bacteria, and very quickly you will see they will be swarming this.
Middle one is the food, right?
Yes, this is a phytoplankton undergoing lysis, let's say. And these smaller ones, like flies, right? You have a dead object and flies. So the same thing is happening in the ocean, but at a much smaller scale. And it's in a very, very complicated environment.
Sir, I have a question.
Yes?
So how they are sensing that there is a food or not?
Okay, so for me as a physicist, I would just say that they have this ability to sense, and turn according to the gradients. If you're a biologist, you will try to go deeper and you will find what exact mechanisms are. I'm not into it. For me, it's too complicated. Maybe some other day. Okay. So as a physicist, all I can say, these are objects which are capable of moving on their own. So they are active and they can sense where the chemical is. Okay.
So the only food source of bacteria is it this dissolved organic carbon? Is
Yes.
it true?
Let's make very simplistic statements like this.
Okay.
So because at the end of the day, you would be seeing that I'm like reducing the problem to
No, just for me to understand this food webs thing. Okay.
kind of, yeah. So dissolved organic matter is at the base, let's say. Okay. So now this collective nutrient search by marine bacteria, collective because there are many of them which are competing, right? So if you are hungry and your brother or sisters are hungry, what about the cooperation? Or there's something like, cake. If there's a cake cutting ceremony and you are like 3-year-old, think about the cooperation. So what are the three components of this problem? Before that, what are the different components of the problem rather? And then how to model each of these components, and do these components interact with each other? Okay. So how can they influence, how will they influence the dynamics of the other component. So there are three components to this problem of nutrition search. One, these are in, let's say, ocean or lake or some big water body. So there can be turbulence. So I'm dealing with turbulent oceanic flow, right? That's component number one for me. Then second is microscale sources of nutrients, right? So there could be at different locations some sources of nutrients to which bacteria would be swarming. And then of course the marine bacteria itself, the bacteria itself. So there are three components that you would like to model as a physicist or as a mathematician: turbulent flow, or flow for that matter. Let's say you want to simplify your life, you will get away from the turbulent flow and you will take some other simple mathematical flow which would allow you to expose the some essential features of the problem. What kind of flows have you considered in this school so far? Have you been exposed to some model flows, mathematical flows? Can you name some?
Roll-me, corner cellular.
Okay, so essentially we will reduce this to one of them, right?
So,
Yes,
how the turbulent ocean is.
that is part of the story. Okay, so collective nutrient search by marine bacteria. Life is not easy, not even for bacteria. So you must be thinking, it's Sunday, I'm here, learning. The same is true for bacteria. Every day, every minute, they're searching for food. Their survival depends on it. Okay, for you, things are slightly more organized. So the motion and reorientation in response to external chemical gradients. That's what they exhibit. They exhibit motion and they reorient themselves in response to external chemical gradients, and they have to worry about survival in a nutrition scarce landscape that depends strongly on chemotactic response. So their survival depends on their chemotactic response in a nutrition scarce landscape. Okay, it's not that, if you look at from our scale, oceans are full of nutrition, depending on how you define it. But for bacteria, it's scarce, spatially scarce. Okay, it's not like, in the entire water is full of things, like at every point. So these marine bacteria have the ability to sense gradients of phytoplankton photosynthesis byproducts and decaying algal matter. So maybe I have qualified the answer for you a little bit more, okay? And they have the ability to locate and migrate to regions of higher concentration. Is this clear so far? Okay. Now, if the life was easy, then things would have been great for the bacteria, right? There is a food source, it goes and eats and relaxes, then again comes back, eats, repeats the cycle. But no, the nutrition search is impacted by turbulent ocean conditions, which are often driven by winds, convection, gravity, okay? And these can modify the distribution of microscale sources of nutrition, okay? Which in turn can influence the movement of chemotactic bacteria. So eventually it becomes a very complex process. And this dynamics occurs at a heterogeneous, small scales in ocean, but it can lead to consequences on a macroscopic scale, on a much, much larger scale. I will show you a diagram where I have, put the length scale corresponding to the bacteria, to the flow, to the zooplankton, just to give you a reference, okay? Yes?
There could be a possibility that this oceanic flow might help this bacteria to.
Yeah, this is part of the game. This is part of the game. It can help, it can, make it difficult, but I don't care. I mean, I'm not emotionally attached to the bacteria, right? I'm a physicist, I'm going to model it. So I will just turn off my emotions, but this process controls the biogeochemistry and productivity of the ocean, and in turn influences the Earth environment. So it's an extremely important part of our planet. Okay. Turbulent flow, turbulent oceanic flow. Now, a huge portion of this problem is the flow. But the difficulty associated with the flow can completely mask the problem that we are trying to study. Okay? Modeling turbulence flows or understanding, simulating is already a challenge. Okay? So you won't be able to simulate the entire ocean just to model, the bacterial response to some chemical. So what would be the size of a bacteria according to you? Microns? And oceans, by the way?
Nanometers.
Huh?
Nanometers.
Oceans, nanometers? Yeah, oceans will extend from nanometers to thousands of kilometers. So would you have a computer to solve that problem? Would you have? No. So you have to be specific depending upon what you want to answer. Okay? Or rather what you want to capture. So scale considerations are extremely important in your problem. Okay? I mean, if you take a flow which is like thousands of kilometers and tens of kilometers of depth, and then you put a micron sized bacteria, I don't know what you're doing. Okay? Then full scale ocean, the lateral extent is much larger than the depth. The aspect ratio is much less than 1. Right? That's what you know about the oceans. So they are almost two-dimensional, quasi two-dimensional, right? Ocean, right? Thousands of kilometers and tens of kilometers depth, right? So effectively two-dimensional. Okay? At the same time, you have to worry about anisotropies which are there, right? Your Earth is rotating, ocean is full of salt, so you will experience, for example, effects of rotation. You will also experience effect of stratification, right? Density stratification. So all that is going to affect the dynamics to some extent. But I cannot afford to deal with all of them. So I will be discarding a lot of rich physics, but at the cost of focusing on my specific problem. So I'm going to ignore much of the complexities if I want to develop a simple treatment of the problem. So what I will do, I will take a two-dimensional turbulent flow for model, and you will see that I will not even keep this promise for the purpose of this talk. But still, when you are considering two-dimensional turbulence, you will have to resolve large number of length and time scales. You will have to worry how a two-dimensional flow field can organize, turbulent flow field can organize on large length scales. So how many of you have studied this or know about this? So have you seen, let's say, ocean, or have you heard that, in ocean you can have large vertical structures, large eddies, right? So the flow is kind of organized on scale of kilometers, hundreds of meters, right? At the same time, there's a lot of physics which is happening at really small scales, right? And at those small scales, dissipation would be very active, right? So you have a scale here and then another scale here, and there's a lot of things happening in between as well. Okay? So even if you have reduced your ocean to a two-dimensional turbulent field, you still will have to worry about this. But again, you can simplify your life depending on what you want to study exactly. Okay? So you have made all this effort, and even with this effort, geophysical flow community will not be happy with you because you just ignored a lot of things they worry about on daily and hour basis. Okay? And of course, you forget about marine ecology, biology, and other complex processes because it's extremely complex. Okay, so now here is physical and biological length scales in ocean, which I have borrowed from this great article. So this is your scale, right? So that's 10 to the power 0, 1 meter, right? So here you can see it's your turbulent eddies ranging from where? microscales, micrometers, up to meters, hundreds of meters, kilometers, and so on and so forth, right? That's your flow. And Kolmogorov flow is, what is a Kolmogorov length scale?
Where Reynolds number is almost 1.
So where dissipation processes kicks in and it starts to make the velocity gradient smooth, right? Where the energy is finally dissipated, right? So that is here somewhere for the oceans, okay? And then you have these nutrients which are like chemicals, which you can treat them as some kind of a scalar field, concentration field, right? For that you can define a similar length scale called Bachelor's length scale, okay? So below Bachelor's length scale, the fluctuations get smoothened out, fluctuations of the concentration field that you're dealing with, okay? Now, where is your zooplankton? Zooplankton is in centimeter to almost micrometer range. And where is phytoplankton? Phytoplankton is 0.1 millimeter to smaller than a micron. And where is bacteria? Our beloved bacteria. Almost at the micron level, right? So this also gives you a size comparison if you see. So there is not one phytoplankton, there is not one bacteria, there are variety of them, of different sizes and all. We will ignore all that, we will just choose some typical size. I will not even mention that. Okay? So this kind of gives you an idea of what kind of length scales you are dealing with. And then you have for the bacteria a motility range, which is nothing but the distance that they can travel during the lifetime of a typical nutrient patch. Nutrients would be coming in patches and disappearing. Okay? Okay, now what?
The bachelor length scale?
So bachelor length scale for in a very simplistic setting, you can take as something analogous to Kolmogorov length scale, where the fluctuations or the gradients in the concentration field get smoothened out, okay. And it will depend on the Prandtl number or Schmidt number, whatever you want to call it, depending on greater than or smaller than 1. Okay. So now what kind of flows or flow environment these objects or marine objects experience? Let's say I dive into ocean, of course I will not survive, but just to experience that flow, you know, or these fishes like whale or some other fish, they will experience these turbulent eddies, okay? So what they will see an aquatic turbulence as a set of interacting worlds of different sizes. You see, many of them may be of the same size or slightly smaller, right? Now if you go slightly smaller into size, like if you become zooplankton, so you basically would be seeing part of this, right? You would be seeing part of this, like layers moving across each other or on top of each other rather. Okay, so at the scale of zooplankton and larvae, turbulence is experienced as a smoother rotation, like they're going and they can get turned, or they will feel the effect of rotation, but they will not see the full eddy or a vortex, okay? Although sometimes as a strong shear as well, okay? Because these organisms are approximately the same size as that of the smallest whirl or eddy that is there. Fine. Finally, you come to phytoplanktons and bacteria, and here you see that they bit overlap in distribution of sizes, right? And what do they see? They see turbulence as a smooth change in flow, speed over space, right? As these organisms are much smaller than the smallest whirl that can be there in the flow. Okay, so that's the flow experienced by different objects there, which are there, and probably this will eat this will eat this, something like that. Okay. Then, I mean, you have a shear flow. I mean, probably you must have already seen today. So shear flow can occur in open water as different layers of water move past each other. That's a very simple definition. And the flow speed changes smoothly over space and can even change direction entirely, as you see here. Okay. Then you come to shear flow close to a smooth surface. So shear flows also occur near solid surfaces because water slows close to a surface, that you know from your everyday experience. Okay. So the shear is felt right down to the scale of the smallest organism in this case. So the smallest organism that is there, that will feel the shear flow. Fine. But if you have a complex topography, let's say of the floor of the ocean, then the roughness, the bottom roughness, can cause variations in the speed as well as direction as shown here. So this can happen centimeter scale or even bigger scale depending upon your interest. Okay, so now let's come to fluid flow modeling part. So first examine.
So can I ask a question? Yeah. So this is something I never really digested, but you know, these microorganisms, they are so small. Mm-hmm. And the length scale of velocities in the oceans, they are so huge.
Yeah, but you have
small ones.
Yeah, but you have processes like which are smaller and smaller. So they are in, you can say, sub-Kolmogorov scale here, if you see.
No, I understand that, but how can I realize that intuitively that, let's say, phytoplankton, how big is it, millimeter?
For phytoplankton, it's micron.
Micron, okay, so
it's like 10 to the minus 6.
How can they even feel the sheer effect of ocean velocities? I mean, if you steer a tank in the lab, I understand it because you're gonna create. But if
to have that level of sensitivity, you have to do that experiment, right, to feel it.
No, I'm just saying.
Or I can just
in the ocean.
I mean, you just scale up the problem.
No.
Yeah, sure.
So basically, so that's the thing about ocean or atmosphere. Like the scales are used. Like it goes.
Yeah.
So it's not like the processes are not happening at the smaller length scale. So of course things are being advected, but you will see that advection may bring things together. For example, if you break down phytoplankton even further to convert them to as a nutrient, that becomes of the order of bacteria. Now what will happen? There will be advection and then there will be so you will see that a small chemotaxis effect can lead to a much it can be very advantageous. If you are passive and if you have small chemotaxis, it can make it.
No, I understand. So inertial effect, I understand. Chemotaxis, not chemotaxis, let's say activities, I can understand. But shear reorientation, like rheotaxis, I don't see how can that play a role in the ocean, or at least the thing is that in oceanic flows, all these scales will be active. So there will be like large length scale, flows. They will just advect it. Means microorganisms will not do anything. They will advect, but not shear.
Yeah, so I'm saying they will just get advected.
Okay, so that's good. Then we agree. Okay.
But somewhere down the line, the hydrodynamic picture will also break down. No?
Yeah, I understand.
You have to be like, you can't go, just keep going. Like you're here 10 to the power minus 10. I mean, an atom is of size. Let's say hydrogen atom 10 to the power minus 10.
So the thing is that in the lab, like basically Raman Stoffer and all, right? Yeah. So they have shown just like how the shear flow and activity.
In the lab, yes, I understand. Because in the lab the scales are so small.
Yeah, but
that doesn't mean it will not exist in the ocean or river or lake. Those scales. Those flow scales will be there, but I guess.
No, they're
there. No, they are, but I think what I'm saying is there's different things.
I agree. Yeah. So which one dominates and all that becomes a different question now. So Yeah, when there are large currents and all, I don't think there will be much effect. But
Because what I'm trying to say is this: let's say you have a huge vortex in the ocean, right? Let's say some of them are even hundreds of kilometers.
Right.
So now, if you basically say, okay, I have a little phytoplankton at this vortex edge. Let's say if you're at the vortex edge at the macroscopic scale, you get a particular shear.
First of all, I mean, these pictures that you see, like they're sitting there where that flow is turning. That's a exaggeration, right? So there could be eddies of smaller and smaller size.
Yeah, but then they detach from the vortex picture. That's what I'm saying. Let's say you have a vortex. Now you
no, but that doesn't mean there won't be any.
Yeah, but let's see. That's the point. So let's say a vortex of this scale, you're going to have a particular shear. But then on the scale that is sort of comparable scale to the vortex, you're going to see this basically shear. But on the scale of phytoplankton, you may have a completely different shear because that's such a smaller scale. It's just their noise compared to the bigger one. So then I was always, when I looked at this paper, I was sort of always confused.
Whether it's a noise or really a shear flow.
Yeah, you're looking at the vortex, but then what's actually felt by the little guy in terms of shear is completely uncorrelated because I can have oscillations on top of the big one. But of course, there are other effects like inertia, advection. They're gonna be there. But the shear itself, sorry, very much.
Okay, no, it's
these are the questions we need to ask.
Yeah.
So now let's focus on. I'm just building the model. Let's focus on the fluid flow. So what we will do, as I said, even though I'm talking about turbulence, and you will see that I will throw out everything out of the window. So what I will do, I will examine the problem for simple flows to gain valuable insights. Maybe one or two flows you may have already covered in this course, and I can avoid see the basic physics by avoiding the complexities which come with the turbulent flow, because there are a set of people who are studying turbulence in the atmosphere, in the ocean, and elsewhere in the neutron stars, right? And if they start worrying about bacteria, phytoplanktons, they will go crazy. So what I will do, I will consider the following two-dimensional flows: cellular flow and turbulence generated by Kolmogorov forcing, Kolmogorov flow, but I will only restrict myself to cellular flow. So do you agree with me on the cellular flow part? So cellular flow that I'm going to consider will have a velocity field given by this expression. So x component would be sin x cos y, and the y would be minus cos x sin y. So what would be the vorticity? What would be the vorticity? How would you compute the vorticity? So you have so what would be the vorticity? How would you compute the vorticity? Vorticity would be simply curl of this, which will turn out to be this, where u naught is the maximum flow velocity. Can anyone remind me about some features of this flow if you have gone through it?
This is there a stream function?
I have not written the stream function. Okay. I've just written the velocity field. I mean, you can get the stream function.
So it's incompressible?
Yeah, it's an incompressible flow. And the flow is, for our purpose, is frozen in time. Turbulent flows are not frozen in time. So I'm going to remove that temporal complexity, and I've reduced the flow very much. So if you see this flow, it will have 4 vortices, counterclockwise 4 vortices, and then it will have a vertical region because of those vortices, and it will have strain dominated region as well. For example, these would be the strain dominated regions, and this would be the 4 vertical regions. So you have counterclockwise rotating vortices, 4 vortices. Fine. Now So now that would be my flow, frozen in time. I have to now model the nutrition field. So what I will do, and this is something I've been telling you, I will treat these nutrients as chemical substances, and then I will simplify it even further. And I will say that for simplicity, assume only one chemical. For example, you are eating dal and rice, right? Lentils and rice, and I say, okay, just survive on rice. Fine. So these nutrients, I mean, that was for humans, okay? So these nutrients will diffuse in the background flow. At the same time, they will be carried around by the background flow. There will be diffusion, there would be advection. And let us represent this chemical substance by a concentration field C. Okay. Now, once you have done this, you will need an equation to describe the spatiotemporal evolution of the concentration field, right? Okay. So the spatiotemporal evolution of the concentration field would be governed by the advection diffusion equation, which is nothing but del C by del T plus U dot grad C, if you want to say it that way. Then there would be a diffusive part, right? And then I have added two more terms to this equation. I was mentioning micro scale sources of nutrients in the ocean, right? So I will have some small spots where the food or the nutrient is being injected, right? That I'm going to model using this FC, which will be my nutrition source term. So I will just have a small patch where I'm going to inject. So lambda is going to be the injection rate, okay? And this xi would be a typical measure of spatial spread over which nutrients are replenished. Okay, a simple model I can take, and then there will be depletion of nutrients, which I will be simply modeling as minus b times some constant time c. Okay, so in the modeling of the nutrition field, I have chosen a time independent flow. Then I will be looking at the flow is time independent, and flow comes where? Flow comes in here, right? So you inject something, the nutrient will be diffusing and advected, right?
Can I ask a question?
Yeah.
So in this, so I suppose that. So first of all, these guys are also precipitating.
Yeah, yes.
No, but is it known what's the speed of precipitation compared to how much they are residing on some stratified?
Actually, I mean, I don't remember the details, but there are specific articles where they have tried to model this. Even in labs, when they are considering it and how much of them is going out of the plane, they take
Just to understand if a two-dimensional model could be accurate, or where are we?
It's more or less hand waving, but you can capture lots of essential features, and that's what the emphasis is here.
Yeah.
So just to look at the patterns and other things that can emerge, that will emerge.
Yeah.
This model allows us to capture. So, but not everything. But what you're saying is certainly happening.
Yeah. And the other thing is, I suspect that basically on these two-dimensional layers, on these stratified layers, the flow is not incompressible because you have subduction, and I have a feeling that this divergence will play a very important role in your prediction.
Yeah, but here I'm kind of...
but it's an easy addition in case.
Yeah.
Okay.
So you would like to know what the steady state would be after the transients have died out, right? You had prescribed a flow, you injected something, and then you waited, like what kind of a pattern for the nutrition will emerge, how the nutrition would be spread out. Okay, so now if CR is the nutrition field that you see, so in the vertical region you see there's no nutrition, but it's, of course, you're injecting somewhere here, right? color bar shows you like this is 0, this is maximum. You are injecting, let's say I chose to inject here, you can inject here, you can inject here, you can inject at the center as well, but that doesn't make sense. So I'm injecting here, let's say, and this is how the nutrition would be distributed. Yes?
So if at particular space, if nutrition is getting injected, then they might get spread or...
This is what is happening, right? So it will spread, I'm injecting, and there is a sink term, right? I'm continuously injecting and something is going out as well, right? So a steady state gets achieved.
So it
Because the
homogeneously distributed, or how the distribution?
So why will it be homogeneously distributed, or when will it be homogeneously distributed?
Because particles are passive, like whatever.
Yeah, but you have a background flow as well, right? So background flow will bring in its peculiarities, right? So
Yeah.
this distribution is emerging because of the background flow. If you change the background flow, this distribution will change,
Sure.
right? So that's why it's not inhomogeneous, because there is a background flow. Otherwise, it will be simply diffusing out from the point where you are injecting. Ultimately, if you consider a huge domain, it will become homogeneous.
But somehow, sir, if I understand something, it is like in the cellular flow or roll mill kind of flow, it should be like homogeneously distribution, right? So since these particles are passive, like whatever concentration or food we are putting.
So this is how the distribution will start. And the thing is that here we have a source and sink term. So nutrients are constantly being generated. So it will reach a steady state. So if there was no source and no sink, then that's it. So if you wait infinitely long time, then it will become homogeneously distributed. But if you have a source and sink, then this is the form. So that's the steady state one is going to consider.
So now this is your steady state concentration field, and for this field, if you compute the gradient, this is how the gradient will look like. These were the spots of vertical regions. So the gradients would look like diverging out from there. If you see the arrows, and arrows also show the strength of the color. They have been color coded and they show the strength. So they are going out from these centers, right? And here these are converging, right? If you have studied this flow in this course, you may have noticed some of the properties. Okay. Then I'm injecting at the center. I'm considering a flow domain which is 0 to 2 pi, 0 to 2 pi in x and y directions. Okay, so I'm injecting at pi comma pi, and the place where I'm injecting would certainly be nutrient rich, right? So the bacteria would like to go there, right? There is a maximum chance of getting nutrients there, right? So what I can do then, around this I can define some source area, okay? Some source area with some threshold. I mean, let's not get into it. So I can define a small area around the point of injection. Okay, that I'm going to call a source area where the nutrients can see food all the time and they would like to go there. Of course, the nutrients are present here, 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 but the concentration is different at different places, and maximum is here. Okay.
What boundary condition are you using for C?
Periodic. Everything is in periodic. Okay, now marine bacteria. So we have covered two components we have covered. One was the flow. So I was saying turbulence, this and that, and then finally I converged to a very simple flow that you can deal analytically, right? And then I said, okay, for the nutrients, I'm going to write an advection diffusion equation, and I will have a concentration field, right? Then the third component is the marine bacteria, and these marine bacteria, I'm going to model them as active chemotactic agents. Okay, and this is where the chemotaxis will play a role. So what are the attributes of these marine bacteria? They are self-propelling, okay? They can move on their own, and they can do a chemical concentration gradient sensing, and that's why I was showing you this profile. Okay. Okay, so if these are active agents, then I need to write down the equation of motion that will determine the spatial and orientational degrees of freedom, and I will simply use a Langevin equation, or rather a overdamped Langevin equation. So this equation will be consisting of three parts. The first part is associated with the self-propulsion, the activity, right? The bacteria is alive, it can move on its own, right? So I have to model that part. So I just said, let's say it's capable of doing a constant self-propulsion, that is, it can move at a constant speed. Its direction can change, and I will specify later. Direction can change. And then, of course, two bacteria cannot occupy the same space at the same time, right? So I'm going to model the interaction between them. I'm going to include the interaction between these bacteria. That I should include in my dynamical equation or not? I should, right? And then they are also being carried around. They're coupling to the flow, right? They're being carried around. So I've just kept the coupling constant as unity. Now let's come to the direction. So the direction is simply cos theta I sin theta I in 2D. And now this theta, I'm going to write an evolution equation for this theta, and there will be three terms for this. One would be chemotactic sensing, okay? So the bacteria would like to align itself along the chemical concentration gradients, okay? And the strength with which it aligns, I'm going to represent by beta. Then it will also experience torque due to the fluid flow, okay, and some noise.
This first term, so grad C and EI both are in XY plane. So I mean, when we are taking this cross, so, I mean this del theta I by del t. So
you take this and take a gradient from here, right?
yes.
Depending upon where you are, you will get the contribution.
But what I'm saying like this, okay, yes.
Okay, so you have three parts to this, okay, and the last part is simply I'm putting a noise, white noise.
I have a question. So, when you say the flow velocity experienced by the bacteria, the assumption at some level is the bacteria is small enough that you can consider it's a single point velocity experience, right? Which means that the scales at which we are considering are much larger than bacteria size. So our grade spacing in some sense.
Yeah.
So in that case, why is it very important to include the repulsion? Because if your grade cell is much...
No, repulsion is like if two bacteria come together, what will you do?
But because your grade size is much larger than bacteria length scales, maybe they can come together. Because you are not resolving the bacteria length scales individually, right?
In this problem, I'm worrying about that, right? I'm prescribing a flow right now. I'm prescribing a flow, and I can go to any extent, right? As far as the model is concerned.
Okay, yeah.
Right?
Yeah.
So then, how are you going to model this interaction?
Can I ask a question? So the bacteria, are you considering them spherical or with an aspect ratio?
Here you can say that small circular disk or of radius RA, if you want to say. Okay. So these agents interact with each other, and to model that interaction, you can introduce a short-range harmonic repulsive force, which you can write down in this fashion, which is nothing but the minus of grad of potential energy, which is simply this. RA is the radius of the active Brownian particle, which you have taken to be the bacteria. Fine. Then the problem throws at you a variety of parameters. First of all, let's say the number of bacteria you're going to treat in this simplified model. Okay. Packing fraction. Okay. The area of the bacteria multiplied by the number of the bacteria divided by the total available area. So in this problem, now after introducing the problem, I have shifted to something that I have worked out a bit. Okay, so the packing fraction that we are going to consider is very small, 0.05. Okay, and I will define a dimensionless number called as a swimming number, which is simply the constant self-propulsion speed divided by the maximum flow velocity, because that is what is there in my prescribed flow, kind of known. Fine.
Sir, why you are choosing this packing fraction 0.05?
I mean, you can play with it, but I will like to keep it small.
Okay.
I mean, if you make the why would you make the packing fraction large? What problems would you encounter?
I mean, it depends if colony of bacteria are there living together, then it can be.
No, but that would be local.
Okay.
What I'm talking about here is the global packing fraction.
Okay.
And you will see what happens locally. Okay. So now I also need a comparative measure of rotational frequencies, that is, chemotactically induced and locally, which is due to the fluid, and I can define a number B, okay, that is going to give me the strength of chemotactic alignment. Okay, so I have introduced a dimensionless number that is going to tell me how strongly these bacteria or chemotactically active agents can align with the concentration gradient. Okay, so B is a measure of that strength. Then I have some time frame in which the bacteria will arrive at the source, food source. Let's say that is tau sub A. So I can compute some average arrival time. Fine. Where tau A sub I is the time taken by the ith agent to first arrive in the source area. Okay, that's the only incident I will consider. Fine. Now, I was setting up the model. I am done with the model. Very quickly, I will show you some results. Now, let's look at the following scenario. You take your flow, you take bunch of these bacteria or active agents, put them at the center of the vortex. Why? Because now you're really playing with the system, and at the center of the vortex you saw the nutrition is minimum. So this guy, if it wants to survive, would like to come out, right? Now, being physicist, what I will do, I will switch off the chemotaxy, I will switch off the interaction and see how the system behaves. So what you will observe, the bacteria keeps moving following streamlines inside the vortex. Okay. Now, this happens at a small swimming number, at a small self-propulsion speed. Now, if it can move at a larger self-propulsion speed, that is as a larger swimming number, it will come out of the vortex and sample the entire flow domain. And mind you, still there is no chemotaxis in it, right? B is 0. Fine. Now, if I introduce the chemotaxis, whose strength I am measuring, or whose strength is given by B, and I take a large chemotaxi, then you see at a small swimming number, the particle kind of spirals out and starts to look for food. It comes out of the vertical region, goes in the strain dominated region. So with chemotaxis, it could sense whatever little could reach inside the vortices, it could sense, align and come out. So certainly chemotaxis helps, right? This is what this very simple exercise shows you, that chemotaxis helps the bacteria to locate nutrients. Fine, you had put it into a nutrient scarce region. The vertical regions were nutrient scarce region, right? But this can come out. Now, if you increase the swimming number or the self-propulsion speed even further, then what happens? The guy comes out and moves in this strain-dominated region and is now able to reach the source area. Okay? So now if you introduce the interaction, you would like to see what happens. So let's say you put the bacteria here, there are a bunch of other bacteria, they're interacting with each other. What you see even at small swing number, it is able to come out and sample the entire flow domain. Okay? And of course at a higher self-propulsion it will definitely come out because it was able to come out earlier even in the absence of interaction. Now you have interaction and chemotaxis both, and that's the problem where we are interested. So what happens? This bacteria comes out of the vertical region, goes in the strain dominated region at, let's say, small swing number, and moves around in the strain dominated region. So this kind of shows you the time as well. So it comes out, then it moves around. At the same time, I have shown the location of all other bacteria simply in red. So at this swing number, for this value of B, you see all of them managed to come out of the vortices and they've started residing in the strain dominated region. So they've started forming small clusters, right? And the video that you saw, there were some clusters, some clusters could form eventually, right? Now here, whatever the clusters are forming, you would like to finally understand what is the physics behind it, what characterizes these clusters. And finally, if you increase the swimming number further, all of them come together at the source of the nutrient and they form a giant cluster, all of them. So it may look like it's a giant cluster where everything is jammed, right? In a small area, all the bacteria have come together and are jammed. And I will show you later, it's not the case. They're not jammed. They're moving inside this jammed cluster. In fact, jamming happens somewhere in these regions. In fact, the jamming or the clustering is such that you can in fact achieve a glassy nature. A glass like this, for example. Okay? It's characterized by what?
Dynamic heterogeneity.
Yes. So you will see that. So this problem that we started with of bacteria feeding on phytoplankton lysis and other stuff in a turbulent environment, which of course we reduce to this, for some specific case can reduce to a clustering which has precisely the same characteristics as this window glass. That's one sort of clustering it can do. The other is this macroscopic cluster, giant cluster that it forms. And this one looks like everyone is there, so it must be jammed. No, this is where they keep moving. In the other one, rather they get jammed totally. Okay?
So there is one time scale, which is roughly speaking the bacteria motion time scale, depending on their velocity and their speeds or whatever, right? And then there's another time scale in your problem, which is how quickly you inject or replenish the source. Now I was wondering, so how these two are comparable in whatever results you showed you? Are they of the same order in some limit? And second, what if you make
So the first one is faster, so that I can maintain a steady state. Yes, and second one?
The first one is faster, meaning the injection rate is less.
Yeah.
Okay, no, all I was wondering was if injection rate goes to 0 or very small, then what do these bacteria do?
So if the injection rate becomes very small, then you would be in kind of where the entire concentration field is diffusing out, right? It won't be sustaining.
Okay.
So, and I'm interested in the steady state for simplicity. Okay. So now very quickly, what I'm doing,
Question.
yeah.
So This, of course, relies a lot on the fact that you place the source at the center.
Yes, yes,
But
yes.
in general, the source, you should maybe have a uniformly distributed source that is transported by the
Yes,
flow.
that's another level of complexity. For example, I can inject, because I was trying to model microscale injection of the nutrients. So what we have done is we have injected, let's say here, we have injected somewhere here, injected here. I mean, for this simple prescribed flow, we have tried variety of configurations, and the glassy phase that I said occurs only when you inject at this point or at this point, which have some special properties which people may have seen in this course. Okay, and if you inject elsewhere, clustering happens, but that's of a usual dynamic clustering and things like that. So it depends a lot where you inject. But what I'm trying to say, this can give you variety of physics, or rather a rich physics. Okay, you can correlate it with other things. Now, what I'm plotting here is something very important, right? I wanted to see whether bacteria can come to the source area or at least pass through the source area. So I'm plotting the fraction of total population that manages to at least pass through the source area. Okay, so this is this curve f, that fraction, versus swimming number. Swimming number characterizes the self-propulsion speed. Okay, so the nutrition search is successful if agents manage to either arrive in, so they can arrive and go and sit there, or simply pass through it. I don't care, but for simplicity I define this as a measure. So there is f versus s. Now you see, as I'm increasing s, this f shoots up quickly, and somewhere around swimming number 0.3 saturates to 1. All of them manage to either arrive at or pass through, right, the source area. But if I increase the swimming number slightly further, you see there is a fall. The fraction starts decreasing, right? And there is a range over which it decreases, 0.3 to 1 roughly. And this dip depends on the chemotactic strength. Fine. Now if I further increase the speed beyond this range, 0.3 to 1, it starts to climb up again, and somewhere around 2, it again saturates to 1. So there is a region where not all of them manage to come. So it means they are happy in certain locations where they are residing, so they might be getting stuck, right? So that's why maybe they are not able to come out. So let's analyze that further. Okay, so what you have so far, you have in fact 3 kind of flow regimes as far as flow regimes for the bacteria or for the particle. Okay, you have a fluid-like phase for S between 0 and 0.3. All of them can come and pass through the source area, come out of vortices and pass through. There is a mixed phase between 0.3 and 2, and you saw that not all of them are able to reach. So what is probably happening, there is a fluid contribution, activity and chemotaxis, they are all competing with each other in that phase. And then there is a chemotaxis dominated phase for S greater than 2, where all of them manage to reach. So if you look at this parameter space, you will see the different regimes that I have shown here. Okay. Okay. Let's very quickly, using very simple observables, try to see what is happening in the flow. So let's look at average swimming speed. I take the swimming speed of each and I simply average. Okay. If you look at that observable, you see as I'm increasing the swimming number, initially it starts to decrease, and for some cases it drops to 0, and then starts to increase and shows some complicated behavior that depends on B. If I zoom in here, you see for different B what is happening. It starts from here, decreases and becomes almost 0, right? In the range 0.5 to 1 for this S, and B let's say 10. right? Similarly, yes.
So if you are increasing the swimming speed, why it is coming to a zero?
That's the mystery, right? I have not done anything. I just, put these guys there and I'm giving them, like allowing them to move faster, allowing them to have a larger self-propulsion. But what I'm finding, even though I'm asking them to move faster, they are going into a phase where their average speed as a whole is dropping to zero. So I'm curious about that, like what just happened. So I'm just trying to analyze that situation, why it is happening. So to answer that, let's look at what kind of flow environment they're exploring, right? So what kind of flow environment they're exploring. So what I can do, I can try to compute the average flow, local flow velocity that they're sampling. So that if you see, average flow velocity that they're sampling or seeing is decreasing as S is increasing, and it hits a minima at some S, which is very much coincident with this. So it seems where this drop is happening, somehow these particles are exploring the low flow velocity regions. That's one. Then I would like to know what further is happening. So I will again look at a very simple identifier or an observable. So I will look at the direction of these active particles with the local flow velocity, right? So first I wanted to know what kind of flow region they are exploring. I got to know that they are exploring the very small or low flow velocity regions, right? Now I would like to see how they are oriented with respect to that local flow. And I can just simply take the dot product of the orientation with the local flow velocity, as simple as that, and average it. And what I see, it drops to a large negative value around the same range, and it's negative. So they are anti-aligned with their orientation is anti-aligned with the local flow velocity. Now if I look at the concentration gradient, exactly opposite is happening. So they are very strongly aligned with the concentration gradient. Okay, fine. Somebody was asking me about packing fraction and all. So it seems there's some confinement happening here, right? They're passing through low phase region and low velocity region. Okay. So what I will do now, I will look at, yes.
So is it fair to say that in the region where they are, where this minima occurs, they are stuck in some sense a local stagnation point, in the sense they are moving forward, but the liquid is moving backwards with the same velocity?
Let's say on an average this is what is happening. Now if you characterize the clustering, that is what the neighborhood look like. You have already characterized the environment. So what I have done, I have taken a very simple, a very complex problem. I have broken into smaller parts for you just to give you an exposure. Okay, and trying to tell you how you would model each part and what you get, and you can get variety of physics depending upon what kind of flow you put, what kind of model you choose for the bacteria or the marine objects. Okay, so what happens if you see this Z, that is the number of agents within 2 RA radius, where 2 RA is the effectively giving the size of the bacteria. So you just immediately looking in the neighborhood of these active agents, and you count the number of active agents around a given active agent, and you plot it versus S, and you see that it starts to increase. And for the largest B that we have, it peaks around 5. That's a large number, right? And then again, it decreases as it increases. But this range is kind of coincident where we were seeing that phenomena. It increases, and then you see for these values of B, there is a second peak. Okay, there is a second peak, and then further decay. So what is happening, I can tell you in advance. You see some clustering here, which is essentially happening in the rest of the strain dominated region, and these peaks correspond to the big clusters at the source. Okay. So what you have observed is a very high local packing. Even though you started with a very, very small packing fraction, overall packing fraction, but there are regions in the flow where you see very high packing fraction. Fine. Now, so what happens? I was telling you here that a cluster can form at the center, right? So the stability of the cluster, of course, depends on the activity, right? The cell propulsion speed that you give to the bacteria. It also depends on the chemotactic strength. And for a given small chemotactic strength, you see first there was a formation of a giant cluster at S equals to 4. Now if you increase the cell propulsion speed, this kind of melts or starts to melt. So you get a small cluster and the rest of the particles are flying around, and if you increase further, it kind of completely melts down. But you can consider a situation where B is large. You would of course expect the cluster to survive for a larger value of the swimming speed. You, as I was saying, you may think that everything is jammed, but it's not. If you look at the single trajectory of the particle, let's say which starts from here, this is roughly the size or the area of the giant cluster. This particle starts from here, okay, comes down, passes through the center, then again moves along the periphery, okay, then again turns, comes along the diagonal, the radius, okay, and again passes through the center and keeps doing this, okay. Now this is something very nice. I mean, if you also know that if you take these active particles and put them in a confined geometry, that can induce rotation. So it's not that, but effectively they are tracing each of these particles are tracing the periphery of the source area. But of course they are passing through the center and all. Now if you can also compute very quickly the mean square displacement, and of course you will see its dependence on the chemotactic strength and the swimming number, and you would observe at small scale they are ballistic, but I'm really interested in the long time scale. So what you will see is that there are cases, for example, S equals to, let us look at the green one, S equals to 0.6. It becomes flat. Then let us look at B equals to 10 and S equals to 7.3. It's again flat. So they are confined effectively, but this particular MSD is not able to tell us what actually is happening, whether they are moving or in what fashion they are moving. Okay. So what I've also done is I have plotted the effective diffusion coefficient for these. So for these smaller ones, for this largest B, if you see, and if I go to a small S region where the problem was happening that not everybody was able to sample the source area, the effective diffusion coefficient is almost dropping to 0. Okay. So now finally let's come back to that S region where F was dropping to a small value. So what happens? These particles come out of the vertical region where they were initially placed because of chemotaxy. They come out, but then they get stuck in another low velocity region. So this pseudo color plot is actually the magnitude of the velocity field, flow field, and the arrows are simply giving you the direction and the strength of the concentration gradient. Okay, so you see they come out and get stuck, and this is giving a sense of time, I mean the distance between these these points. So there is a slowdown here. So if I zoom here, this is how it looks. If I zoom further, this is how it looks, and if I zoom further, it looks like this. Okay. So finally the takeaway points. So a simple environment can give rise to complex regimes depending on the slow regime, chemotaxis. Okay. So this whole exercise was to illustrate this. So you can have a fluid-like phase where everything is good. These marine particles or bacteria move around, or rather, I should say, active particles move around freely. Then there can be a mixed phase where there is an effective trapping. So this phase can show you glass-like features, and if you characterize, for example, the local swimming velocity and with the fluid velocity, you can see that there is a 1/f noise which is kind of linked to this. So the whole thing is very rich. You started with you were thinking of dynamics of the bacteria, how nutrition uptake will happen, but if you start analyzing, it gives you a very rich physics. And finally you get a chemotaxis dominated phase and you get a giant cluster. Okay, so that's part one. I will take only 5 or 7 minutes, Anupam, if you allow me.
Please.
Okay, so that's part one. Again, I have not done any justice to the actual problem. As a physicist, I just made the problem smaller and tractable and tried to tell you that you can use what you have learned so far, here or elsewhere. You can simply analyze the situation. Now, so orientation dynamic, again, I will not completely do this part. I will only just give you a glimpse. So orientation dynamics of phytoplanktons. Actually, these phytoplanktons you were looking at, which were dying and the bacteria were coming. So that's another part of the problem. Okay. So you have these phytoplanktons, variety of phytoplanktons, different sizes, lengths, which if you recall from the scales that I had shown you, that there is quite a distribution. Now if you can take one of them, and what you will see that there is a unequal distribution of mass, so that you can have a so that you can say that these phytoplanktons are bottom heavy. Okay, so center of mass is slightly below geometric center. Okay, and they do a different kind of taxis, which is called as a gyrotaxis, which is biased swimming caused by a balance between gravitational torque and the hydrodynamic torque. So this gyrotaxis happens in a marine environment, and I can say marine flows are rarely still. They are turbulent. I mean, you will encounter turbulence, shear, upwelling, downwelling regions, and so on and so forth. So this gyrotaxis makes motile phytoplanktons cluster in specific regions of the flow. Okay, so earlier we were talking about bacteria, now we are talking about phytoplanktons, and what you can see is that these phytoplanktons, when they're clustering, they can form thin phytoplankton layers which are centimeter thick, but they can be kilometers wide, and it can be disastrous for the marine ecosystem. Okay, that's why people were interested or are interested in understanding these. Okay, and of course turbulence comes with patchiness, so this all becomes interesting. People have observed bioconvection, preferential sampling of the flow by these. Okay, that's again some examples that people have studied, like dynamics of these gyrotactic swimmers or phytoplanktons in shear flows, how they can get trapped. Fine. Now I don't know if this picture is clear to you, but if you see these green patches, so it's Oman, it's Pakistan, somewhere it's here India, and you see the scale on which this is happening, these patches forming. So these patches influence the marine ecology to a great extent, and these patches form because of pollution. So we dump, let's say, fertilizers which have phosphorus and nitrogen, right, which become nutrients for these phytoplanktons, and they start to grow, populate. Okay, and this is happening over a much larger scale, and the dynamics that I was talking about, chemotaxis, is happening on a smaller scale. And if you recall the statement that I made, what is happening at a smaller scale can lead to effects at a much larger length scale. And this is where the flow physics of the underlying flow and other things come into picture, and that's why people are interested in modeling these things. And people have done variety of things, and for variety of flows, for example, here is one, then you see a convection-like pattern, a roll-like pattern, right? Okay, so ecological and environment relevance, and so the primary production, photosynthesis controls light and nutrient competition, carbon cycle affects carbon cycle export to deep ocean. Okay, fisheries, phytoplankton distribution affect higher trophic levels. I mean by higher trophic, like fishes and all. So if it forms, the one which are there, for them it's disastrous. Okay, so you can have these harmful algal blooms, and gyrotaxis contributes to bloom formation and its persistence. So people are interested in understanding its formation and its consequences. Okay, so quick history. So 1950s to 70s, biologists observed that many flagellated algae show bottom heaviness. For example, Chlamydomonas dunalela. Okay, and they did experiments that showed that these guys orient upwards in still water, and they have a mechanism to maintain, which is a mechanism to maintain light exposure for photosynthesis. So this is all it started, I mean, slowly, where biologists came first. Okay, then came again biophysicists, or you can biologist/physicist, and who looked at the suspension of swimming algae, and they saw emergence of bioconvection patterns due to upward swimming of these guys. Okay, then came a physicist or a mathematician who formalized the hydrodynamics, like you are learning in this course. Okay, and they introduced the gyrotaxis model, combining the cell physiology, that is bottom heaviness, and the fluid dynamics. Right, this is where we are slowly headed, I mean. So what we do, which the story I will not complete, now I take a fully turbulent field. Okay, again a local section maybe somewhere. I'm still not doing, that aspect ratio much less than 1. I'm just doing homogeneous isotropic turbulence locally, and trying to study how these phytoplanktons orient in a turbulent environment. Okay, so I solve this Navier-Stokes equation on a computer while enforcing incompressibility condition. So you can see the snapshot of the velocity components ux, uy, uz on some plane. Okay, but this is a full 3D field, and you can derive this. You can derive the equation of motion for the orientation vector of the phytoplankton just by looking at the torque balance. A swimmer experiences two main torques, one gravity and fluid. So you can then neglect the inertia and then introduce gravitational torque, which you can write like this. Okay, and eventually you will arrive an equation which is given by this. Okay, and this constant b that you see is the reorientation time. Let's say the phytoplankton is kind of misaligned with the z-axis. Let's say if you're looking at with the vertical, the time it takes to align with it, and it will be our measure of alignment tendency. Okay, so you can write down an equation of motion for the orientation vector that you get by looking at the bottom heaviness or the cell physiology and the hydrodynamics. Then again, Anupam told me that you have been exposed to these paths. So antisymmetric part of the velocity gradient rotates the swimmer, so it will have this term, and symmetric part of the velocity contributes to the rotation with different aspect ratios. So I can consider phytoplanktons that are spherical, spheroids, and rods. Okay, so I will have these terms. So finally, I can write down an equation of motion for the phytoplanktons. I will have, x dot, which is equals to the background flow plus the orientation vector with the swimming number here coming, which is nothing but the self-propulsion divided by the Kolmogorov velocity, because I'm dealing with a 3D turbulence. And this is the full equation for me that I can solve. These I can solve while I'm solving the flow velocity. Here I'm not considering a stationary flow. I mean, I'm not considering a stationary flow or a time frozen flow. I'm considering a flow which is evolving in space and time, a fully turbulent field. So every part is active here, and I consider objects like this. Okay, so different aspect ratio, gamma is 0 for a sphere, for a spheroid 0.5, for rod 1, and I also consider different stability numbers, you know, a measure of the reorientation time, how much it takes to reorient. Fine, and I will just end with this, even though it's not the full set of results. So if they are slowly reorienting, so it means they're experiencing all the directions which I have plotted here as a px, py, pz, where px, py, pz are the components of the direction vector along x, y, and z. And you see it emerges as a spherical kind of an object. Okay, so each point is giving you the orientation vector. Now if you have a fast reorientation, you see most of them align along the vertical, that is the z. And you can go on and compute, you know, the tendency, alignment tendency, or rather its PDF, whose dependence on, let's say, Reynolds number, the aspect ratio, the reorientation time. I will not go into it. You can then further try to dig deeper if you are really into it. You can try to see how they align with the eigenvectors of the strain rate tensor, vorticity, and there are some surprises here compared to, let's say, you are dealing with just passive guys. You can compute autocorrelation to see how long they remain correlated. And here again, you can, on top of this reduced modeling, you can have further reduced model. You can reduce this 3D problem to 2D. You model your flow by you take your flow as a random flow and you just come down to 2D. So this term will drop out if you're considering sphere, and you simply solve it. And what I'm trying to say, even this reduced model gives you some of the essential features that you get from the 3D simulation. So that's the power of modeling. That's what we end up doing. We cannot solve a fully turbulent environment in which we will not be able to model, let's say, all the micro sources of nutrition and all the bacteria or varieties of bacteria. So we will start developing models to make the problem tractable. And of course, this will have drawbacks like the one I showed you. So I chose a flow, a cellular flow, where I injected at a particular point. Even that injection gave me variety of clustering of patterns, but that relied heavily on where I'm injecting. Okay, so next is you would like to solve it for a full 3D thing to understand. Okay, thank you.